Today, it's my pleasure to introduce the next speaker, who's Jessica Turner, who I've pretty much known for like forever, right? Feels like in a good way. So I guess we met a long time ago when you were in California, heading up the FBurn, organizing the FBurn activity, and then we worked together at the Mine Research Network for a few years, and then you jumped over to GSU, and then I moved over to GSU because I just had to work with you again. So, and so Jess is an associate professor here in neuroscience and psychology. She is a core member of the Trends Center, and she does a ton of stuff, and she's been really active in organizing the ENIGMA activity, specifically related to the schizophrenia working group, but in lots of different contexts. And that takes a ton of work, and it's been making a huge impact. And so I think you'll see some of that in the talk today. And so she's been a big proponent of data sharing, of collaboration, of open science, and all these good things that are happening now. And hopefully the world's catching up to where she is. So, so Jess, without further ado, I will it off to you, and it's great to have you join us.
Great. Thank you so much for inviting me. I guess I'll go ahead and share my screen and bring it up and go ahead and start it now. I cannot see the chat window once I'm in, because I'm now in full screen mode. So hopefully, Armin, I'll task you with watching the chat window. And if somebody has questions, just either tell them to break in and stop me, or we can certainly wait afterwards. And I do intend to make a PDF of these slides and put them up on the Slack channel as well, so that if there are citations or other references going by, you can catch them afterwards. And I want to encourage you guys to join the Slack channel for that. So thank you so much for inviting me. Those of you who know about ENIGMA, who've participated in ENIGMA, or who have heard presentations about ENIGMA before, stick with me. There will be some new information a little bit later, but I do want to give the background on what ENIGMA is for those who may not be so familiar with it. So ENIGMA stands for Enhancing Neuroimaging Genetics Through Meta-Analysis, and it's really the brainchild of Paul Thompson, who is currently at USC. And back in 2009, everybody was very excited, certainly in the schizophrenia world, but also in other research domains, and doing imaging genetics. And we all thought if we could just get to the point where we can all collect data together, or we can all collect, 1,000 subjects, we can probably find some real breakthroughs in the imaging and genetics world. And it turns out, of course, life is not that simple. And it's very hard to find anything with only maybe a few hundred subjects along the way. And Paul's big idea was basically to say we're all collecting structural imaging when we collect these imaging studies. And we're all collecting genome-wide association data or genome-wide scan data. So what if we tried to put it all together? Because most people were not interested in the structural imaging. Most people at the time were very interested in the functional imaging and in looking at genetic effects on cognitive processes and cognitive processes as they play out in functional MRI. So a lot of the structural imaging data was just sitting there doing nothing. And Paul said, look, why don't we give it a try and actually work on the structural imaging that people are less protective of, perhaps. And so his big idea was really, don't ask everybody to send their data to a central repository or to put them all in a central school or give up the data to somebody else, but to leave the data where it is. I think somebody is typing. I'm sorry. Is everybody muted? Thank you. I'm not outside. Okay, so his idea was not to aggregate the data, but to leave it where it is and let everyone do the same analysis on their own data. So do a prospective, a planned meta-analysis. So not look at the literature and see what's out there and try to put it together from what's available in the literature, but literally to work with data that might otherwise not have been analyzed, have an agreed upon analysis pipeline, and then do a meta-analysis. So you're not asking people to give up their valuable genetic data or their valuable imaging data, just do an analysis and send the summary results along for a meta-analysis. So in 2009, he and I think Barbara Frank and a few others sent out emails to everybody they knew who might have genetic data and imaging data and said, hey, would you be interested in trying this out? And they actually got a fantastic response, so that their first paper literally pulled from cohorts all around the world. This is the original cohorts that were and sites that were participating in ENIGMA that participated in the first imaging genetics paper that was published in 2012. It was not a fast process, and it was all volunteer. People just saying, sure, I can do this, I can do this in my free time. Just let me, let us interact with everybody about it and publish the paper together. By 2014, so just 5 years later, it had been organized into a number of working groups focused on disease and imaging questions. And these was, obviously the distribution of groups around the world in 2014. And most recently, we can see it's grown into 51 different working groups, most of which have more than one chair. They're often co-chaired because, again, not funded. We're doing this largely volunteer, representing 31 disorders and multiple other efforts across 400 institutions in 43 countries. And I noticed that he's actually, he was so happy when he got one in Russia, and we got lots of people in Japan and Korea as well. So all around the world, people are participating if they have imaging data that they're willing to collaborate on. So the first paper, well, let me actually say a little bit more about the organization because it helps to give a bit more background. The heart of ENIGMA was, of course, the ENIGMA GWAS effort to say, how do you put imaging together with 1 million or 2 million genetic data points? And from that, they were very successful in looking at cortical and subcortical measures, as well as DTI. The EEG methodology has also grown up, as well as how do you put genome genetics together with EEG measures? What do you do with epigenetics, copy number variations, and different approaches, as well as collaborating with other genomic groups? That also had to develop hand in hand with protocols for doing these kinds of image processing. So besides the huge effort of dealing with the genetics, how do you actually deal with the images in a way that is robust and repeatable across sites? And of course, the subcortical volumes is, again, the first one out the door with that development, the first sort of test that was put together with the genetics, followed by cortical and the rest of the subcortical areas. Now we have hippocampal subfields. Again, EEG has a working group that's trying to figure out how do you meaningfully analyze EEG in a similar and comparable way across different institutions with different equipment, et cetera. And there are different methods also in development here. Then, of course, once the first ENIGMA GWAS was actually published, the individuals who were participating in this, the researchers who were contributing their data and their analyses, realized that many of those data sets had come from the studies of the genetics of various disorders, with schizophrenia, bipolar disorder, and ADHD really being the primary contributors, the big bulk of the contributors to the ENIGMA GWAS. And therefore, they were starting to organize. So that's how I got into organizing the ENIGMA schizophrenia working group, which has since spun off many other working groups, the bipolar group, the ADHD group. And you can see over time, as new groups have sort of expressed interest, they've developed into their own working groups. PTSD, autism, anorexia has been charging ahead, and I'll say more about some of these newer groups a little bit later. Then besides disorder, there's also groups working on how do you look at the non-disease imaging data. So we can look at changes over time, lifespan development, lateralization. These are three groups that have formed to really study what can we see if we're looking at thousands and thousands of the healthy controls that were collected as part of these data sets. How can we make sense out of that? So the methods of what that ENIGMA really depended on in the early days was very dependent on the fact that there were software that was in common use. Freesurfer was a software for doing structural image processing and cortical and subcortical segmentations and measurements and extracting the volumes and the measurements on the gray matter images that has been around forever. I think Freesurfer was probably started, I want to say the early 90s, but it may have even been earlier. And they had standardized on a directory structure that had been the same at this point for 20 years. And so everybody who worked with imaging data, who worked with Freesurfer, was familiar with how to handle data that was arranged in these orders. And it was very straightforward, somewhat straightforward, to write scripts that could then say, if you have done a Freesurfer analysis, take these scripts and it will basically walk through all of your subjects and pull out the cortical volumes and subcortical volumes that we want to analyze, and then write it out as a CSV file. And then we can all agree, a lot of effort went into agreeing on what is the actual analysis that you want to do. What are the regressions that you want to do okay, I don't know what I just did, but I'm hopefully not going to do anything too exciting. What it is that you want to actually do with the data, which can then be automated and then the results can be sent back. So whether you're just analyzing case control differences or you're analyzing a standard genetic analysis, it can all be automated. Everybody does it exactly the same way. And then the results are sent back to a central analysis team, who then proceeds to have to do a great deal of data curation and double checking to make sure everything happened appropriately and somebody didn't miscode their gender or miscode their ages or handle missing data in the wrong way or something like that. But then there's a meta-analysis step that happens along the way. Let me see if I can figure out how to stop annotation here. Okay. So the first analysis that was published from ENIGMA actually successfully was applied to 13,000 data points or individual data points across, I don't remember how many different sites that contributed structural imaging analyses along with their genetics. And they focused first on the hippocampus and intracranial volumes. They said, let's keep it simple. Just two volumes that we're really interested in, the hippocampus, because everybody's interested in the hippocampus, and overall head size or intracranial volume. This gives you an example of, it took 3 years to get it all together and working, but they did actually get the paper out looking at the genetics of the hippocampus and the intracranial volume. And for those of you who aren't familiar with these kinds of images, this is basically a zoom in on chromosome 12, a particular genetic region, and showing where in the analysis and the meta-analysis, we're seeing genetic variation seeming to be related to hippocampal volume. And along this axis is, of course, the p-value, but at the negative log 10, so that small p-values are up here and non-significant p-values are down here. So we're looking at an area of chromosome 12 where we're seeing some hits, some areas where genetic variation relates to hippocampal volume. And one of the reasons I particularly like this paper, and I'm hoping you all can see this, this is the same graph as before in 12q24.22. And it's this is a locus that's related to the expression of the gene TESC, which is important in brain development, cell differentiation. And of course, that's all the genetic informatics question that comes, okay, if we find something, what does it mean? Why might it be related to brain volume? But I'm always fond of these kinds of figures because this is really where the heart of ENIGMA is in the meta-analysis. So each of these data sets had maybe a few hundred subjects, together. They add up to the 13,000, or maybe across this, that's 13,000. But each of them only has a few hundred subjects, and none of them, this is the effect, per allele, basically the change in hippocampal volume, basically the effect size for that genetic variation on the hippocampal volume. And none of these would be published, right? You couldn't publish that. But when you put them all together, you can see that there is a consensus in the effect, so that the meta-analysis actually pulls out the consensus effect and says, yeah, there seems to be evidence here that there is actually a genetic effect. And similarly, they then were working with a number of other groups to do a replication. So not only did they do a meta-analysis, they did a replication of the meta-analysis, and in the replication, they also showed, again, no significant effects in each individual site, but a consensus effect that supported the initial result. Very exciting stuff. For all, it was, basically saying, okay, here's, one little area that seems to be related to hippocampal volume. They then could go on, and in 2015 published the full subcortical analysis, looking at all of the subcortical regions and not just the hippocampus. They replicated the hippocampal result, and turns out the putamen actually had much stronger genetic effects than the hippocampus did, and the paper is obviously available for more exploration in terms of exactly what those genetics were for the subcortical volumes. And then most recently, last year, the full cortical measures analysis, the GWAS analysis of the cortical regions, was published, this time now with 50,000 subjects from around the world. It gets very difficult to find reviewers for these papers, as you can imagine, which is why it was up in bioRxiv for a long time. To actually get that reviewed and published, because so many of the imaging geneticists were actually part of the process, part of the project. But it's been very exciting to watch ENIGMA really grow again, to just scale up and scale out. And because in those initial analyses, they discovered that, so many of the groups had a common focus on a particular disease, we formed the schizophrenia working groups, the major depression, bipolar, ADHD, and then other groups have formed since then looking at various disorders. And each one of them could say, okay, step back from the genetics. Right? Let's just step back from the genetics. Let everybody who has imaging data and wants to participate join in, whether they have genetics data or not. And we can start looking at what are the case control differences in each disorder and really nail that down. And what are the effect sizes? What's our best estimate across a worldwide conglomeration? What's our best estimate of the effect sizes of case control differences across these different disorders and within and across different disorders? So schizophrenia, this is our cortical effects. Basically, schizophrenia has massive effects. And when you've got almost 10,000 subjects in the study, pretty much the whole brain is affected significantly, though the strongest effects are obviously in the temporal lobe and the frontal lobe, as we would expect, and less so in the sensory motor areas and the occipital lobe. Bipolar disorder looks very much like schizophrenia, as we would expect. They had slightly fewer subjects, but they're seeing the same general pattern with the motor cortex being relatively spared. Major depression, hardly any effects at all. It's actually very hard to get consensus in a major depression study in where the effects actually are, because it's such a diverse disorder. ADHD is much more uniform. Obsessive compulsive disorder, again, frontal and temporal, but not the superior temporal areas. Autism, etc. Lots of comparisons. And of course, now the different groups can actually start comparing across disorders, as we are wont to do, to say what's really different, what's really the same, where do we see comparisons, and what might that mean for the distinctions across these disorders, the mechanisms underlying these different disorders, etc. So we're now seeing, in a sense, meta-analyses of meta-analyses, in the sense the groups are getting together to say, okay, what's the same and what's different as we compare ADHD and autism and things like that. That's sort of the next step. Now, not every analysis is gray matter, right? So far, I've just been talking about structural imaging. There is also other pipelines, the diffusion tensor imaging pipeline and the resting state pipelines have both been developed, applied, and published. And of course, the question in developing a pipeline, what does it mean to have actually developed a pipeline? You're trying to reduce the effects of heterogeneity across the samples. You're trying to make sure you have a pipeline that is robust, as error-free as possible, as easy to implement as possible across sites. But at the same time, you want to maintain your effect sizes so that you've got a homogeneous estimate of your effect size, even if the samples are very different in how they were collected and other concerns. So what Neda Jahanshah, Peter Kochanov, and others did when they were developing the DTI pipeline was really test whether it maintained heritability of the white matter measures. So they worked across several data sets, large data sets from around the world, that had family structure in the data to say what is our estimate of heritability. With the data sets that they had, they could share them, so they could do both an aggregated or mega analysis and also analyze each data set separately and do a meta-analysis and confirm that, sure, the mega analysis is a bit more sensitive, but the meta-analysis showed the same pattern, and so therefore it was an acceptable pipeline for use, and that is the ENIGMA DTI pipeline that everybody has been using since then. Very promptly, the schizophrenia group published a DTI paper, the bipolar group published a DTI paper, the major depression group published a DTI paper looking at fractional anisotropy largely in those disorders and saying which tracks actually show the largest effect size. And then the Japanese ENIGMA, which is their own consortium that's very similar to how ENIGMA works, but all within Japan. They basically took the ENIGMA pipeline, applied it in their data sets, and did a quick look to see whether or not the correlations are the same. Basically, are the effect sizes correlated across their analysis and the ENIGMA worldwide analyses? And within schizophrenia, it's very high, bipolar disorder, slightly less. And again, major depression, very heterogeneous samples there. And the correlation is not as high, but it's still in the same direction. So this is not heritability the way it was in the original pipeline development. This is actually looking at case control differences and showing, again, these are finding reliable differences and repeatable differences across different groups. The resting state fMRI pipeline was developed in a very similar way. Let's look at heritability. Let's make sure that the pipeline that we come up with, which is open access, easy to get access to, make sure everybody has the software. It's not dependent on somebody's internal pipeline that they only works in their data. Does it actually both reduce the inter-site differences and maintain the heritability estimates so that we think that it would be usable in an imaging genetics kind of analysis, and that has been published as well. And there are, of course, new pipelines still in development. The most recent two are actually the magnetic resonance spectroscopy ENIGMA working group. They are actively looking for collaborators who have MRS spectroscopy and have MRS expertise to join the discussion in what a pipeline that would be generically best use across multiple sites would look like, and how would you process data that is heterogeneous and how it was collected in order to really extract the common effect. And similarly, Henrik and others have taken on the task of saying, how do we look at task-based fMRI, and how do we combine task-related analyses across different sites? And of course, the issue with functional MRI, with task-based fMRI is everybody's got a different task, right? Everybody uses different timing. Everybody uses slightly different conditions. Even within a lab, you might change your experiment a little bit from one study to the next. So they're starting with, I believe, the NBAC, which is a very common working memory task, and saying, if you've got NBAC data, come talk to us and join us in our discussions of how do we take into account, just within this one paradigm, the differences that people have had and how they've implemented that in order to extract a consistent consensus estimate of effects within fMRI. So that's a huge effort, very exciting to watch. And not everything is a meta-analysis. Several groups, I think starting with the attention deficit group, found that mega-analyses were actually easier for them, and enough of their participating sites were willing to share data that they could actually create centralized databases of shareable volumes from Freesurfer analyses or the outcomes of the DTI pipeline, so that they could do a mega-analysis and have all the data in one place rather than doing a meta-analysis. And most recently, Joaquim Rajua published, actually literally just out now, in Neuroimage, a paper where he was looking at data from the ENIGMA schizophrenia group. Some of the groups could share their data, and so he could do both a meta-analysis and a mega-analysis at the same time, and he was specifically looking at the combat algorithm, which I know some of you have worked with already, and arguing that combat actually did seem to increase sensitivity over the meta-analytic approach. And his scripts are openly available, implemented in R. And if anybody would like to recreate that or apply it to a different disorder, he's happy to help that along. So ENIGMA has been going now for 10 years, and there were a number of papers out last year on sort of the 10-year retrospective on ENIGMA. And as of this month, there is a special issue coming out in human brain mapping that is really pulling from a lot of the more recent developments in ENIGMA that I wanted to talk a little bit about. Okay. So some of the papers that are being published currently, there's about 18. I think there's even more than that. They're going to have 2 special issues, but the ones for September cover both updates from various working groups as well as updates on various of the methods, talking about mega versus meta analysis, reproducibility in some of these questions, and also empirical analyses looking at depression, schizophrenia, addiction, etc. So there's really quite a lot going on there, all based on ENIGMA or reuse of ENIGMA approaches. So something about the ENIGMA diseases, some of the newer groups that have come online, have started working, and are publishing in this special issue are the epilepsy, where they have to really deal with the fact that not every epileptic patient is the same. The focus of the epilepsy may be starting in a different part of the brain. How do you compare across one group versus another group? Stroke is facing the same problem. They know physiologically that the stroke patients are very different. How do you actually combine that to get a consistent meta-analytic approach across different sites and different studies and different recruitment techniques, etc. The ENIGMA Anxiety Group is very aware of the hierarchical umbrella of anxiety disorders, looking at generalized disorder, phobic disorders, social anxiety disorders. How are they going to organize that? How are they going to pull that all together and really look at that? So the newer groups are really diving into the heterogeneity underlying their disorder diagnoses, and really that's going to make for some very interesting results that I think move the fields forward. I wanted to say a bit more about this one, where Peter Kochanoff and co-authors have taken the ENIGMA findings, particularly from the ENIGMA schizophrenia publications, and said, how can we use that to take it from a meta-analytic, a massive kind of study, large-scale analysis, down into the kind of study that you might do at your own research site. So he was using the ENIGMA schizophrenia findings to develop a regional vulnerability index. So what is a regional vulnerability index? A regional vulnerability index is calculated if you've got your imaging study and maybe you've got cases and controls and they're age matched across each other. You want to estimate your brain measurements for each subject and get your mean and standard deviation for the healthy population from your sample. So in this case, for example, we might have a Freesurfer data set, and this is an example of the Freesurfer cortical parcellations. Each color is a different region, and you would have for every subject the thickness, for example, the cortical thickness from each of these regions. From your healthy controls, then, for your study, you get the means and the standard deviations, and then you normalize every subject relative to the healthy controls or the healthy population in your sample. So instead of having the absolute thickness, you now have a measure of the deviation from the healthy controls for each subject, because even your healthy controls, some of them might be a little bit bigger here than average or a little bit smaller there, and your patients might be a little bit bigger here and a little bit smaller there, whatever it happens to be. From the ENIGMA schizophrenia papers, we have sort of the worldwide international estimates of how do patients with schizophrenia deviate from healthy controls. How are they different? Where do you expect the largest effects to be, accounting for age and sex and intracranial volume and mean thickness and whatever possible analysis you might like? And what you can do in this case is calculate the regional vulnerability index by correlating the individual subject, correlating the individual subject's Z-scores with the Cohen's D scores across the region. And a higher correlation means the the person looks more like a case than a control, and a smaller correlation would be more like a control. And a negative correlation would be more like, the opposite of what was found in the literature for the patients with schizophrenia. So what Kochanoff et al. Did was take their own data set where they had 120 cases and 78 patients with schizophrenia, and they had a lot of measurements on their patients. So in the paper, they also look at cognition, they look at symptom severity, etc. I just want to show you the duration of illness findings. So they grouped their patients based on whether they were early in their disorder, so within 5 years of diagnosis, whether they were mid-range, maybe 5 to 20 years, and whether they were late in their illness or over 20 years. Now, this does not necessarily mean you're very old. Remember, many patients develop schizophrenia between 15 and 25. So they might be, they might be 35. They might be 38. This is not an elderly population. They're just people who've been living with a disorder for at least 20 years. So what they can do at that point is look at how the regional vulnerability index differs across patients based on duration of illness. So your controls, basically your regional vulnerability index hovers around 0, mostly negatively correlated with what you expect from the patients. Their patients overall are rather positively correlated with what you see from the ENIGMA schizophrenia, looks very similar. And if you look particularly at the subcortical and the white matter and the multimodal, they figured out a way to put the gray and the white matter measures together and come up with a correlation using both of them. The early patients, the ones who are closer to the onset of their illness, look less like what you see in the ENIGMA patterns than when they are getting to be more intermediate or later. So there seems to be an increasing similarity to what's been seen in the literature based on where along their stage they actually are. The reason I find this particular paper very interesting is because they are working with the kind of data set that you might work with locally to say, how do you translate what we're seeing in the literature from these massive studies down towards a study that you could actually do, and what might that mean as you're heading into working in the clinic? Could you actually use these regional vulnerability indices to predict symptom onset or the onset of the illness, or who's going to actually develop the illness? Can you actually use it to predict anything you like, treatment response, etc.? Can you dig into this to look at the genetic effects? Do people through the controls who look more like the patients who have a higher regional vulnerability index, does that correlate with polygenic risk score? There's all sorts of questions that you can ask as you sort of incorporate these kinds of approaches. And he's not the only one. This is one of the papers from the special issue where the substance use group is looking at their patterns of finding and explicitly comparing it to the previous other disorders meta-analysis to say where do we see an interaction or an interplay or a similarity between addiction and the various kinds of addictions that they've had to look at and the psychiatric disorders, and what does that mean in terms of underlying mechanisms, reward circuitry effects, you name it. There's all sorts of places you can go with that. And within this special issue as well, Vince has a paper with Elizabeth Block really looking at the data sharing, which is a tension that we always deal with, the tension between meta-analysis and mega-analysis. Can you share the data? How do you leverage the ability that people have to work with their data to really make these large-scale collaborations work? What are the pros and cons of aggregating the data versus decentralizing it, etc.? So that's actually everything about ENIGMA, and I'm hoping I've given you a taste of what we can do and the newer developments with ENIGMA, more than just, oh, hey, there are cortical differences across these disorders, what the value is to the rest of the research community. So basically, I wanted to highlight that large-scale data collection, management, sharing, analysis, meta-analysis are becoming more common, more possible, easier to do, even more so. The analyses are becoming more powerful, more sophisticated, more nuanced. We're really digging into the heterogeneity of many of the disorders that we know is there. to say, if you can have these 10,000 subjects, 15,000 subjects, can you actually start to tease it apart in a way that really identifies consensus effects? Combinations of meta and mega-analysis are being used. We're trying to be flexible in the approaches that are being applied. Obviously, structural, functional DTI analysis pipelines are publicly available. You can go and get them. And newer pipelines are being developed, and they are looking for people with expertise to join in. And novel approaches to decentralized analyses are becoming available. And obviously, Vince's team and the CoinStack platform are interacting with ENIGMA to try to help facilitate those kinds of analyses, so that even if you wanted to do an analysis that traditionally requires all the data be in one place, if you want to do a machine learning technique, if you want to do an ICA or one of these multivariate kinds of approaches, traditionally we've had to have all the data in one place in order to do an iterative analysis. And that's becoming less true as time goes on, which is just wonderful. The initial results from all of the data sets are public and can be reused. And there are comparisons across disorders and diagnostic boundaries. Obviously, there's the HBM special issue where you can get all the detail that I've had to skip over in this presentation. But I'm hoping I've sort of whetted your appetite for interacting with these groups, interacting with the larger ENIGMA consortium, and trying to think about how these results can impact the particular disorder or cognitive process or technique that you are interested in working with. How do you join ENIGMA? Traditionally, have imaging data will travel. Initially, most of the ENIGMA groups basically wanted people who are affiliated with the data set, either because you're the PI, you collected it, or you played a key role in collecting it, and it's okay for you to share it and work with it. Join up, bring the data, help out. More recently, we've seen cross-disorder applications, so we get people proposing projects. They don't have schizophrenia data, but they have a method, and they've worked with somebody in bipolar disorder or addiction or ADHD, and they want to apply the method in a new disorder. That's becoming more common. And more unusual, but still occasionally done, is somebody with neither a method nor imaging, but they have a question, they have a hypothesis. They won't, maybe they don't have any relevant data to ask the question, but they can make a connection with someone within the relevant ENIGMA group who's willing to either let them try it out on their data and then sort of sponsor them to make a proposal to the full group, etc. So there's lots of ways to get involved, and please feel free to reach out to me or others who are chairs within ENIGMA for more information on that. I wanted to highlight everybody who's been involved, which is largely these folks down here. This is the chairs group from last year in Rome. So this is, again, maybe half the ENIGMA chairs who were able to get there. And each one of them represents a working group with hundreds of other people involved. So that's not all of ENIGMA by any stretch of the imagination, but quite a few of them. And this is my lab, so many of whom are involved with ENIGMA or ENIGMA CoinStack kinds of analyses and are helping out with that sort of approach. And Vince's lab as well, which has been developing CoinStack and some of the decentralized algorithms, which are going to be coming more and more into play. So thank you for your attention. And I hope I haven't completely blasted through people who were trying to say something, but I'm happy to take questions.
Thank you, Jess. Okay, so do we have any questions? I
saw the chat. There are highlights in the chat window, but I haven't seen what they said.
They're so just, Mukesh says good morning.
Oh.
So I have two questions, if you don't mind.
Sure.
So one question is about the regional variability index. I was wondering if they normalized for data within each scanner, or is it across all data set? And my second question is about ENIGMA. Does it mean like the data is not freely publicly available? How does it work if someone doesn't have the data, but they want to use the data?
Thank you. Okay, well, that's two big questions. First, in the regional vulnerability index, they're basically developing the regional vulnerability index or applying it to one data set that was not in the ENIGMA group. So they're normalizing it within that data set. Okay, the ENIGMA data set the ENIGMA results were not normalized. That's a meta-analysis analysis across multiple data sets.
So I'm confused. So they normalize their own data set, then how do they, and they do not normalize the data we have in ENIGMA, right? And
That's
how
right.
they're comparing them?
They're literally, they have the effect sizes from ENIGMA for the same regions that they have z-scores in their own data. And they're for each individual subject, they would correlate that subject's z-scores across regions with the Cohen's d effect sizes across regions that ENIGMA's made available. Okay.
Thank you.
So hopefully that clarifies things. So you're not comparing the Z to the D, but you're just correlating it, sort of, rank order, to see how similar they are. Then within ENIGMA, no, ENIGMA is not a data sharing consortium, right? The whole idea is that the data can stay largely where it is. So it is not, you can't go to ENIGMA and say, give me your data, because in many cases they're behind firewalls where the institutions have said, according to our institutions or the way the data was collected or whatever, or the national rules that have come into play, we're not allowed to share that data. So if you want to, for example, run an analysis on a subset of the ENIGMA data, we have methods for making proposals. So you can basically propose to the ENIGMA bipolar working group or the ENIGMA autism or the ENIGMA HIV data set or whatever working group, whatever you want to do, to say, I would like to, propose this kind of analysis. What is your group's rules for doing that? And then you interact with the chair of that working group to say, okay, how do we actually make this happen? Because different working groups have different ways of going about it.
Thank you. Thank you.
Mm-hmm. So, Jess, what would you say? Maybe you already mentioned this in your talk, but what would you say is the biggest advance that has come out of ENIGMA since it was started?
What is the biggest?
Put you on the spot there.
Yeah. No. Well, the problem is there's a lot to choose from. I think there's a huge sociological effect, right? That basically says, oh, look, people will actually work and play together nicely, right? 2003, we were having serious doubts about that. So I think that's certainly a huge step. It's basically the fact that we've gone from one imaging genetics study in 2012 to 50 different working groups, all asking questions about their methods, their diagnoses, their what is it that's going on. I think that in itself, that mushrooming effect is a huge impact. I think it's really changed the frontier for imaging, large scale imaging. It's not the only thing that's changed the frontier in imaging, but it's one of them. I think some of the cross diagnostic comparisons are really key, because when we published, for example, the schizophrenia results, they were not surprising. You could have predicted them from the literature, from the findings, everything else. We could have predicted those. What we couldn't have done was make a definitive estimate of the effect sizes. It says, if you look across the entire world and everybody analyzes their data the same way, so you removed all the heterogeneity that comes from using this pipeline or that pipeline or this covariate or that covariate, you can actually get a really consistent estimate of the effect size. Now, how do you tease apart medication and other things? That's a different issue. But to be able to say this is the definitive set of results, you don't have to ever do this study again for schizophrenia, bipolar, and all these other working groups, I think is just a huge sort of flag in the sand that says, okay, we can now move forward from this. Now we can move forward in terms of teasing it apart to say how much of this is due to medication, how much of this is due to genetics, how much of this is due to environment, et cetera, et cetera. But at least we can say, here's what we expect to see.
Thanks.
Hey, Jesse, this is Yiping. So I have a question regarding the, data, quality control. Since ENIGMA involves so many groups worldwide, so how do they, normalize the data? Do they follow the same protocol, to do the data analysis or data collection?
Nobody. Well, all of these are legacy data. So the data was collected as it was. So whatever they used whenever they were collecting the data is, was the best they could do at the time, generally speaking, right? Everybody tries to collect the best data that's, on the best scanner that's available at the time that they collect the study. But clearly, some of this data was collected in 2006, some of it was collected in 2018. It's going to be very different in terms of the actual imaging protocol, which is one of the reasons everybody's very interested in the mega versus meta analysis, because meta analysis is very forgiving of the different ways you collect the data. Particularly, I know the gray matter analysis is the best. And looking at, for example, 1.5T versus 3T analyses, if I scan you in a 1.5T and I scan you in a 3T, and I'm sure if I scan you in a 7T.
Losing you, Jess, a little.
Oh, sorry. Can you hear me?
Yeah, your audio is kind of cutting out.
Oh, sorry, talking too fast. What I was trying to say is we know that the brain measures that you estimate from a 1.5T, a 3T, or even a 7T are going to be very different, right? If the estimate that I get of gray matter thickness probably will vary, even though your gray matter thickness hopefully doesn't, but the estimate will. However, the effect size for schizophrenia versus controls really is not that much affected by a 1.5T to there didn't appear to be an interaction between scanner and diagnosis effect or the effect of interest that we're looking at. So the meta-analysis is very forgiving for those kinds of issues. So that said, each pipeline also tries to develop quality assurance techniques to say, look at your data, visually check your data. What constitutes a good image versus a bad image? What if you've done your Freesurfer analysis or your DTI pipeline, what are the checks that make you say this subject probably shouldn't be included in the data? So that is a good portion of pipeline development, is trying to figure out exactly what those things are. But usually it's not specific to your data collection. It's more to the outcome of the processing. What raises a red flag to say this value is out of whack, don't use it.
Okay, thank you.
And that's why the spectroscopy people are going to have a fun time trying to figure out exactly what the quality assurance should be and what the processing pipeline should be.
We got a comment from Enrico. Says, impressive talk. Appreciate the opportunity to submit new questions or ideas to the ENIGMA consortium. Even if unusual, it would be a great step forward for those on looking at complex interactions among genetic development, aging, and potential modulating effects.
Absolutely. Absolutely. There is a brain aging group, and there is a genetics group, and the genetics of brain aging, I'm sure if it isn't a group already, it probably will be. And that's definitely the kind of thing that Paul Thompson, as sort of the spider in the middle of the web, he gets a lot of emails from people saying, how would I get involved with X? How would I, what can I do about Y? Is there a group that's interested in Z? And he's happy to point you in the right direction. Because there's no rules about who starts a group. It's literally who's got the interest, who's got the time, who can actually pull it together.
Yeah, thank you very much. I don't know the possibility to submit a question or ideas is really very interesting. In my opinion, for example, is a little example in my experience. I really interested in cognitive reserve paradigm and ideas in also in psychiatric and neurological disease in general, and even in normal people or healthy subjects. So the possibility to test or try this concept, this idea on a very large, huge data set or paradigm like ENIGMA, it's very, very exciting. So it could be. Yeah.
It's a very slow process. I will say this, that it's a slow process to go from a specific question to a full analysis, because again, you have to come up with, what does it mean to look at cognitive reserve? How do we operationalize it? How do we operationalize it across different studies that collected different cognitive measures? What is it we actually need to, and then what's our analysis? How do we get people to implement it, et cetera? But yeah, that's exactly what this kind of thing would be for.
Yeah, on the other side, there's no great study on, for example, cognitive reserve or other basic questions in neurodegenerative disease, for example, or even starting with the methodological analysis point of view. So we have to bring together these two words. So thank you very much for your presentation.
I think, Jess, it would be kind of cool to, as we've been sort of working on the coin stack efforts with ENIGMA. So imagine if you sort of operationalize some different, like a sort of a set of measures, right? And a set of imaging modalities, and then you offer up a suite of analytic tools, and you know, everybody's kind of agreed, and then you make this sort of an open consortia that people can join via the CoinStack tool, and they can run analyses that they want and work together. And so the hard work's done up front once, right?
Right.
And then obviously everything's hard work, but you know, you then can, you know, very quickly run analyses and ask specific questions of those measures in the context of different kinds of imaging and
absolutely. And that's the kind of thing that Neda Jahanshad's trying to facilitate in keeping. She's trying to actually just build the portal, a portal that sort of says, here's all the analyses that are being done across all the different working groups in ENIGMA, because in many cases we end up reinventing the wheel, right? Because somebody does a cerebellar analysis over here and somebody else develops a cerebellar analysis over there. And even within schizophrenia, we've run into two people trying to do different thalamic subnuclei, you know, protocols, and they've had to sort of work together to harmonize it and compare results and things like that. So yeah, that's, I think working with that kind of huge list would help a lot to say, not just within the data sets or the working group that I'm interested in, but in other working groups, is somebody already doing this? Can we really leverage what's already been done? It would be absolutely fantastic.
Yeah, I'm even thinking of something even more automated where you can kind of, as an engineer, right? You don't want to talk to people, right? So let's just provide a place for someone to go and click some buttons and get some results.
The huge social advance.
Yeah.
This huge social advance, you're just going to nullify it.
No. It's part of it's enabling. It's enabling all of it, right? So
no, exactly. Right. No, exactly, because we want to make what we can as easy as possible.
Right. And there should be credit to everyone, you know, obviously ways to give credit for everyone for this. So yeah.
Exactly.
Cool. Okay. So and Armin just pointed out, I guess another good thing with decentralized is you can share features, not the original data.
Oh yeah.
So if there are privacy issues, it can sort of help with that.
Yeah. I actually got an email from somebody. One of my friends in Minnesota, actually, who was like, what is the point of doing all this decentralized stuff? Because don't we all just have to put it in the database anyway? And I said, NIMH funded stuff, yes, has to go in a database. NIH stuff probably has to go in a database or has to be shared. Not true in the rest of the world. And so if you really want to do international consortia, you've got to be willing to work with the GDPR or the Japanese government's regulations, and the Chinese government will never let data leave its borders. So you have to be able to figure out ways to use these decentralized approaches.
And we touch on that a little bit in that paper with Tanya White.
I'm sure, yeah.
But there's more that should be done there. So excellent. Well, thank you. Let me see. Did I get all the comments? I think we did. Thank you, Jess.
Well, thank you for inviting me.
Yeah, and thanks everyone for joining. And Armin, are you still on?
Yes.
What is our next talk? In a couple weeks, right?
1 second.
Yeah, I know.
2 weeks. Is in 2 weeks, and it is Dr. Simon Eikhoff.
Simon Eikhoff.
Oh, that would be
September 25th.
So there we'll dig deep into psychiatric disorders, I'm sure. So yeah.
So the title is Leveraging Biological Knowledge from Brain Mapping to Predictive Models.
Excellent. Okay, well, great.
Thank you.
Well, thanks everyone. Be sure to keep in touch via Slack, and we'll talk to y'all soon. Have a great week and weekend.